Good morning, students. This is a class continuation for the alkaloids. Regular class is on Saturday, but due to a staff member want to take a leave, I'm just coming over to this class. This is today about, we are going to discuss about two drugs, Vinca and Rauwolfia. And Vinca, it is a very common plant which we can see in our roadsides and in our city, in our town also it's possible, and also we used to use this plant as an ornamental plant, and also it can be seen in our garden. So usually when you are coming to study about any crude drugs, the botanical source is very important, and also its family should be specified very clearly when you are starting the details about any plants when you are describing. So this is Catranthus roseus is the botanical source, but usually when you are writing botanical sources in pharmacognosy, we used to write a dried leaf, dried root or dried stem obtained from the definitions will be clearly written like that, which part we are using as medicinal purpose. And but here we are using the whole plant, that is why directly we are writing the name. So Catranthus roseus, commonly known as bright eyes, Cape Periwinkle, graveyard plant is usually found there, so it's why graveyard plant. Madagascar periwinkle, old maid, pink periwinkle, rose periwinkle, and you can see the flowers. Sometimes the colors are different by different places, and also it has many species also, but most of them, they are very important drug. Family Apocynaceae, it is native and epidemic to Madagascar, but grown elsewhere as an ornamental and medicinal plant. It was found it was formerly included in the genus Vinca as Vinca roseaceae, and synonym, that is why we are continuing, that is Vinca roseae and Periwinkle roseae and Lochnera roseae, Spatch, and Ammocalis roseae. And chemical constituents, when you are coming to chemical constituents, very important. You have to write chemical structures also very important. While studying itself, what is the difference between certain constituents, when you remember, you can write very clearly specific things which will give you more clear picture for how to write the structure, because it will be somewhat complex, but anyhow it is easy for you to write it down. And vincristin and vinblastin, and vincristin, you can say both are there. They are chemotherapeutic medications used to treat several types of cancers, are found in the plant or biosynthesized from the coupling of alkaloids catranthine and vendolin. So there are two molecules are there, catranthine and vendolin. These both together, they are coupling together and giving vincristin and vinblastin, a slight difference in them. So that's what very easily you can remember. The structure will looks very big. So because there is two structures, catranthine and vendolin will join together and it will give vinblastin one structure, and at the same time they also again they will club together and they will give vincristin, a slight substituent different. We'll see the structure, and so that you will be understanding about that. The newer semi-synthetic chemotherapeutic agent Vinorelbine, used in the treatment of non-small cell lung cancer, can be prepared either from vendolin and catharanthine. So, either vendolin and catharanthine from the natural source, the structure can also be produced to Vinorelbine is a drug very much useful for the small cell lungs cancer. It is also can be used for production. From the Vinca alkaloids, leurosin is in both cases via an hydrovinblastine, and rosinidin is an anthocyanidin pigment found in flower of Catranthine roseus. So this is also one pigment which is found in the Catranthus roseus flower. So it is also a chemical constituents when you are studying. So you see the structure of the vincristine and vinblastine. Actually what I want to say is, here even slightly it was not seen, especially this catharanthine and as well as vendolin structure together we can see in this vincristine and vinblastine. So vincristine and vinblastine, they are having only a small change. The R will be changed with methyl group in, if it is methyl group is present, then it is vinblastine, and if it is aldehyde group is present in R in the place of the second indole position N group, it will be vincristine. So this is an indole alkaloid totally. So catharanthine is an indole alkaloid. Vendolin is also as a indole alkaloid. So two molecules of this indole alkaloids, they both are having a side chain. Just you can see in indole alkaloids, they have multiple nitrogen inside the nitrogen structure, one or two times. When you see how many carbon atom and actually what are the substituent which is present, it is easy for you to remember actually. So they both of them are joining together and it gives vincristine and vinblastine. So that is why we are told that it's clubbed together. The vincristine, vinblastine is formed from especially catharanthine and vendolin. So one or two times if you know this, if you see the structure, it will be easy for you to write it down in a paper and then you can study this. It's very important to write structures. Without structures, the pharmacognosy teachers will not give you any mark when you are writing some chemical constituents. Uses, especially as we know that it is highly used for mainly cancer drug, it is anti-cancer drug. Even yesterday also we were talking about this Vinca. It was the bundle of when you are taking the plant, even if you take 1 ton, then only you will get 3 grams of constituents can be isolated. So very minimum quantity is present, but they are very effective, very effective. So the uses, you can see the species have long been cultivated for herbal medicine and as an ornamental plant. In Ayurveda, Indian traditional medicine, the extract of its root and shoot through poisonous and are also against several diseases. In traditional Chinese medicine, extracts from it has been used against numerous diseases including diabetics, malaria and Hodgkin's lymphoma. Hodgkin's lymphoma is usually blood cancer. It's blood cancer, which is very critical condition. Even they can be treated with this vincristine and vinblastine, which is present in Vinca roseaceae. And many of the Vinca alkaloids were first isolated from Catranthus roseus, including vincristine and vinblastine, and used in the treatment of leukemia and Hodgkin's lymphoma. Catranthus roseus can be extremely toxic if consumed orally by humans. Chemical test answers all general tests for alkaloids. And adulterants, allied drugs, substituents, it is necessary. You might have studied about adulterants and substituent in the last semester when I am the teacher which have taught you. So you may be knowing that adulteration, intentionally adulterated and non-intentionally adulterated, sometimes for the business purpose, sometimes it is by means of the collector without knowing different uh, parts of the plants may be included, or sometimes the different plants also can be included without constituents. So, but in substituent they will be having a constituents, it can be substituted, may not be as such as the original plant, they have other constituents also may be present. Other Catharanthus species such as Catharanthus longifolius, Catharanthus trichophilius and Catharanthus lanceus are known to contain Vindolin type of alkaloids. So now we are coming to the next step of the plant, which is Rauwolfia serpentina, the family Apocynaceae. I did not mention family, simply I have put Apocynaceae. Better to use as family Apocynaceae. So both of them are Apocynaceae family. Both of them are having even indole alkaloids. This type of relationship is there, but we are not studying about what is the cultivation collection and microscopical characters. All we did not study for Vinca, because in your syllabus they said we need to study more detail about Rauwolfia. So that's why I included in Rauwolfia, but I have deleted some of the part in Vinca. So definition for Rauwolfia consists of dried root and rhizome of Rauwolfia serpentina. So root and rhizomes are used for the medicinal use. That's why it is written it's a dried root and rhizome which is obtained from Rauwolfia serpentina. A small shrub found in India, Pakistan, Java and Thailand. Common name Rauwolfia Indian snake root. That's why you can see Rauwolfia serpentina. It is a Sanskrit name. When you see serpent, it is a snake. So why it is used as a serpentina? Why it is called snake root? Because the shape of the root looks like snake, and so that it is told as Rauwolfia serpentina. Cultivation, how to cultivate and collect is very important for this Rauwolfia, because it is cultivated even, because it is having more useful for hypertensive patients, and even it has useful in psychiatric activity. It has used as a tranquilizer. Many activities are there. It has neuro effects are there. That is why it is having some of the critical usage or more in pharmaceutical, in the health field. That is why even in pharmacy we are also having more importance by this plant, and it is a traditional plant. Very long back it is used. It is not a new plant which is identified recently. It was a traditional plant like morphine, opium. This is also a very old plant, and it is traditionally in Indian system of medicine like Siddha, Ayurveda. In all this system, especially the main two systems which is available in India, they are using this from the beginning. They have standard preparations also. It grows spontaneously in tropical forest, temperature 10 degree to 40 degree centigrade, which are humid in summer at an altitude up to about 1200 meters. Rauwolfia plant cannot tolerate temperature below 50 degrees centigrade. From cultivation, rain must be enough. For cultivation, rain must be enough and abundant in summer, or it should have good irrigation. Also, watering is very important. Irrigation is very important for Rauwolfia. And Rauwolfia grows well in clay, acidic pH 4 to 6 and well manure soil, and plowing must be deep for facilitating the development of roots. As we are interested to take roots and rhizome, it needs soft soil, soft atmospheric of soil condition that will make the roots to develop faster and good quality. So propagation, now how to propagate, because cultivation you need the propagation, you need the seedlings, you need to have many places should be covered with this plant, means propagation is carried out by planting seeds, root culture, and even it can be done by tissue culture or stem cutting method can also be done. But seed propagation gives better yield of root in spite of the fact some seeds are weak in germination. But anyhow, seed propagation is the best one. So therefore, cultivation of Rauwolfia is usually carried out by seed propagation. Immersing them in saline eliminates sterile seeds. So you can see collection, how they are doing collection. Anyhow, you have to collect from the underground. So they are collected in what time? It is October to November, after hot and dry period. The Indian pharmacopoeia, in Indian pharmacopoeia, collection of root of 3 to 4 years old plant is mentioned. But in culture it is found, culture means in the tissue culture, it is found that root of 2 years old plants are equally good. So that's also can be used for the propagation, for the planting, the plant and cultivation. For collection of root plant are dug out, aerial parts are removed and roots are separated. Roots are washed and dried in air till moisture is about 10 to 12%. Roots should be stored protected from light. It is very important, you have to see the moisture content when you are storing, because if the moisture content is more, automatically the mold will grow, that is bacteria will come and attack, and so that your roots and rhizomes which you have collected may be completely deteriorated. It may not be useful for the medicinal purpose. And you can see now macroscopical character. So students should know, in this junction we are going to see in this semester, that is regarding macroscopy, microscopical character. Better to have a practical section, because practical makes the student to know about what is the reality. So that is very important. But anyhow, in online class we were unable to do practical, but when we are coming to the institution, we will proceed with the practical. At the time we will take transverse section, we will say the microscopes which are more commonly used in pharmacognosy lab, we can use it. Usually we are using 2 microscopes very commonly in the lab. One is decision microscope, another is a compound microscope, but we have even electron microscopes are also available. Compound microscope you might have used even in the microbiology for finding out the motility of the microorganisms by means of using oil immersion, and 100x you might have used. But here we are using 10x, 20x, 45x, the maximum we used to use for our purpose, and to find out the cells, and also take a thin section, mounting in the slide, and also using the reagent, clearing reagents, and also staining reagents like fluoroglucinol, which all things when you are doing, you will find very good slides can be prepared and can be observed in the microscope. So what is the difference between the decision microscope and compound microscope? Very commonly using in the lab is decision microscope is used to see the macroscopical characters, that is external character. It has only a 5x eyepiece and a small table, small table in the slide in the particular instrument, that is decision microscope, where we can keep the part of the plant, or we can keep the drug which is available. Root can be kept, and you can observe what is the external structure, color, and you can observe fractures and what all the external appearance and roughness, all these things can be observed and measured also. Macroscopical character, that is called decision microscope. It has only an eyepiece with a small table, a glass table in the instrument itself. And if you see a compound microscope, it is coming for the microscopy. It has two, that is, you might be knowing that it has an eyepiece and as well as an objective lens and a telescope, and also it needs a telescopic arrangement which will have a multiplication of very magnified and which can be seen by means of your eye, by means of seeing through the eyepiece. So that is what we can see in the difference between the decision microscope and also a compound microscope. Anyhow, when you are coming to the lab, it is very important to know about various parts of the microscope and also how you have to use it, and it's very important to know how to use the light and magnification and diaphragm. All these things will come when you come to the lab. I will explain you clearly. Now we can go to the macroscopical characters which I have mentioned in this slide. You see that external features of the root and the rhizomes are nearly similar, but rhizomes can be made out by the presence of small central pith. So rhizomes will have a pith. Usually, it's also similar to the stem part. So rhizomes, which when is present, you can see even the pith, that is the young xylem, can be present in the pith. That is the pith, which if you want to study about even the microscopical character or macroscopical character, when we talk about the plants, it is necessary for you to know about basic botany, what you studied in biology. So you have to refer that basic botany, what all the small parts will be available in the stem and root and the leaf. These all things you might have studied little. That is why you should know what all the cells which will be present. And also drawing a diagram of the histological characters are very, very important in pharmacognosy. Neat pencil diagram. No pen diagram, nothing will be allowed. When you draw a diagram by means of pen, immediately it will be scratched off. It's a wrong procedure. You have to use pencil and rubber for always when you are coming to write an exam or writing any type of procedure for pharmacognosy. And a drug consists of mostly small pieces which are 2 to 15 centimeter long and 3 to 22 millimeter diameter. Pieces are cylindrical, slightly tapering and tortuous. They are tapering and tortuous. You can see the picture how the drug which is taken, and the whole root I have just shown, it looks like a snake. That is why it is Serpentina, Rauwolfia serpentina. And you can see the outer surface is grayish yellow, pale brown or brown. Fracture is very short. What is a fracture? Fracture means usually dried material of a crude drug or anything. We will know to know the external morphological character. When you break, what will be the fracture size? Sometimes it will be polished, sometimes will be powdery, sometimes will be very, very sharp needle like. So like that you can make it. When you break, what is this one is short. When you break, it is a short like some wood. When you break like sandalwood or something small, sandalwood, when it will be hard, small one I am telling. If you break, it may be short. So that's what fracture is short, and fracture surface shows yellowish to brown bark and dense pale yellow radiating wood. It is dense yellow radiating wood with 2 to 8 annular rings and occupying nearly 3/4 of the diameter. Odor, odorless, and taste is bitter taste. It's very bitter taste will be seen in the bark, in the root. And you can see the microscopical characters. As a root, it has many characteristics of the parts which we can see, that is microscopical character TS, that is transverse section. So when you come to the lab, you may be knowing what is transverse section, what is RLS, radius longitudinal section, and radial longitudinal section. These type of sections will give you an idea about the cells, how it is present in the part of the plant. So TS, that is transverse section of the root, present a circular outline with a typical stratified cork, that is a cork cells. You can see in the first below me there is a diagram which you can see stratified cork cells. They are dead cells which you can see cork cells. Root stem, when you see the plant, always roots will be having sometimes a bark, a cork will be there, outermore dead cells will be there, stone cells, scleroid cells also will be there, but here it is less. only cork cells are there. And they are arranged, you can see the following are the tissues seen in the periphery of the center, and it is also you can see other secondary features also seen in this structure. So the first one is periderm. In periderm you can see, which is shown, that is the cork cells, straight cork cells. There is two layers are very cork, phloem and phloem. So when you see phloem, that is phloem is stratified, consists of alternating bands of smaller subsidized and unlignified cell up to 8 to 10 row in radial depth. Larger subsidized but lignified cells up to 5 to 7 row in radial depth. So that's what we can see in the cork cells. And also phloem cells. Next one is phloem cells, indistinct but is seen as a narrow layer of thin layer cells. You can see a phloem. Indistinct but is seen as a narrow layer of thin walled cells can be seen in the phloem. So you see, next one is a phloem. Phloem is 5 to 7 layer immediately below the phloem cells, is arranged in the radial rows, whereas away from phloem cells is oval and has intercellular spaces. And phloem contains abundant of starch. You can see in the picture also I have shown that starch grains, and with an triradiate hilum, that is a pointed area, and a typical twin prism of calcium oxalate crystals can also be seen. Even in the picture also we have shown that phloem contain calcium oxalate crystals and starch grains in the cells with small dots can be drawn. You have to draw this diagram and you have to practice and to make it very clear how to draw and how the cells can be done, polygonal cells, how it should be drawn. It's 1 or 2 times if you draw, then only the practice will come. And secondary phloem is transverse by conspicuous medullary, that means conspicuous medullary rays, that means transverse by conspicuous medullary rays. You can see in the picture the cells can be identified. They are arranged in such a way because it is transverse, and when you see in a longitudinal you will see a long pipe like, but in the transverse you can see this in a shape of polygonal shape cells, but they are arranged in a series, and the phloems contain of sieve tubes, companion cells and phloem parenchyma. Starch grains and calcium oxalate prisms occurs throughout the phloem tissues. You can see they are also containing this calcium oxalate prism. Calcium oxalate crystals are also present. And in secondary xylem you can see here in the picture I have shown that it is a vessel. Vessels are, you can see it is the xylem, secondary xylem. It is also transverse by well developed medullary rays. Xylem consists of vessels, wood fibers, even fibers are also present, wood fibers and lignified parenchyma. What do you mean by lignified parenchyma? Lignified means it contains lignin. When you can identify when you take a section and mount and a clearing fluid, and then you are using a staining fluid. There are many staining fluid. In that if you use fluorogrucinol, the fluorogrucinol will react with lignin and they will show that pink color, so that you can see xylem vessels and xylem fibers can be easily seen very clearly pinkish color due to presence of lignin. That is why we are telling lignin, that is lignified parenchyma and fibers are present. The vessel appears round, polygonal or at time radially elongated and occurs inner single or in pairs. Xylem fibers appears as rounded and polygonal structure with thick lignified walls. Typical oxalate prism and starch grain resembling those of the phloem and and phloem occurs freely in wood parenchyma. Medullary rays, it runs radially from the center of the cortex through the phloem. Rays in the xylem region are lignified, pitted, and are 1 to 5 cells wide, although unicerate rays are prominent. In the phloem region, the ray cells are not lignified. That means in the phloem region, especially when it comes near to the phloem region, the medullary ray cells are not lignified. So you can see clearly they are colorless after mounting with the even coloring or the staining fluid. Starch and typical calcium oxalate prisms crystals are also in the medullary ray cells can be observed. So usually if you want to go for more identification, starch grains, when you are using iodine solution, it will give bluish color can also be observed. Purple bluish color can also be seen. So next one is that chemical constituents, alkaloids, or here it is present, here also it is present indole alkaloids. We have seen in Vinca also they are also indole alkaloids, even Rauwolfia also indole alkaloids present 1.5 to 3%. Weak basic indole type 7 to 7.5 pH. Reserpine group, it has reserpine, resinamine and deserpidin. This is the three reserpine group structures and also the constituent which is present. And tertiary indole alkaloids are also present, which is Ajimalin group, Ajimalin and also Ajimalicin. And strong basic anhydronium bases also present in the pH of 11, and also another group which is responsible for various uses, that is serpentine group, serpentine and serpentinin, and also tonin is present. So these three are serpentine group, and there are two Ajimalin group and three reserpine groups. There are nearly this, there are many others are there, but these all of them are having therapeutic activity and pharmacological action which are more useful for the medicinal use. You see the structure now which I have shown. It is easy for you to study. You study according. This is that is deserpidin. It is just hide by means of this camera. You can see that in the deserpidin it is, and reserpine and resinamine. So resinamine and reserpine, there is not much difference. You can see if you see the structure is similar, only you see up to this structure, only thing is the side chain which you can see carbonyl group, there is CO, C double bond O, and then the substituent of methyl dimethox, that means trimethoxybenzene group is attached. Here also the trimethoxybenzene group is attached, but only thing is you are having one more 2C double bond CH double bond CH group is attached, and alkene group is attached in between these two substituents. You can see, observe that, and that is why it is coming to cinnamic acid. It has a cinnamic acid derivative. It is a cinnamic acid derivative in between the structure when it is clubbed. And also you can see deserpidin, you can see that it is also having a very slight difference from reserpine, very slight difference. Only OCH3 I put on round there. Only the OCH3 in reserpine is not present in deserpine. So these three of them are having similar structure which you can easily study. And also serpentine, alsontine and also serpentinin. Serpentine, you can see the same structure which is not having the side chain. You can see there is a 3-6-membered ring which comes same like reserpine, and also alsontine also same like reserpine. There is not having much difference. Only thing is they have only an alsontine and also serpentine, they have only an isomeric change is there, and also there is they have an steric, that is they are having only a they have only the rotational difference. But you can see that serpentine is having double molecules. There is two molecules of serpentine is joined in their indole alkaloids. You can see indole structure like what you seen in vincrestin and vinblastin, how they are joined, two molecules. One is vendolin and another is catharanthine, how they are joined together. Here also we have two molecules of structure, two molecules of serpentine joined to serpentinine can be easily identified. And the last one, there is Ajimalin group. It is also similar, Ajimalicin and Ajimalin. So Ajimalin is little only thing in a complicated structure of the third ring structure is connected with the first one directly, and the third that is the lactone ring is missing from the Ajimalicin. That's why Ajimalin is Ajimalicin. You will get from Ajimalin to Ajimalicin. So these all are having similar biosynthetic pathway route which comes from tryptophan, amino acid, and also it has to enter into shikimic acid, and also it has a complex derivative of many structures which is obtained by this shikimic acid pathway. And so you can see Rauwolfia uses. Now when you come to the Rauwolfia uses, Rauwolfia is used as hypotensive and tranquilizer. So it is an anti-hypertensive drug. You might have studied in pharmacology when you are studying first ANS itself, they might have told about what is the action of Rauwolfia. And reserpine, being the main alkaloid, is responsible for the activity and is used in anxiety condition and other neuropsychiatric diseases. So you can see sedative, it can be sedative and calm down the activities of excitement. Reserpine group can be used and stimulates the central and peripheral nervous system. Ajimalin groups can be used, and the decoction of the root is used to increase the uterine contraction in difficult situations. Sometimes in the labor, in the delivery time, it can be given for an easy delivery and told to induce the labor pain, can also be given as a decoction of the root, is also given in the olden days and also in traditional medicine. The extract is used in intestinal disorders and also anthelmintic, worm clearance, bitter tonic and febrifuge. It can be also used as various uses. And substituent and adulterant for Rauwolfia serpentina. The following species of Rauwolfia are substituted for genuine drug Rauwolfia ometeria. This can be distinguished by official drug based on the sclerides which are present very abundance here, but you cannot see more sclerides in original Rauwolfia serpentina. Rauwolfia canadensis here again the stone cells, it is also similar like sclerenchyma. Stone cells are present, but the characteristic stratified cork cells in the outermost cell of the root is totally absent. So by that you can identify. And another one, the Rauwolfia micrantha, shows both stratified cork and stone cells and stone cells. All these do contain reserpine and others less important alkaloids in small quantities. And adulterant, sometimes adulteration, that is all substituent. Adulteration, Rauwolfia densifloria and Rauwolfia paracensis do not contain reserpine. It doesn't have reserpine. They do not have stratified cork cells and stone cells. That is the end of this today's class, a small class, and we will continue with the other drugs in the next class. Thank you very much for being